Hello students, I am Əliyeva from Department of Pathological Physiology. We continue our distance lessons. The topic of this lesson today is cell injury. Cell injury or alteration is a typical pathological process characterized by disturbances of the intracellular homeostasis, impaired cell function and cell structure. The cell, which is influenced by pathological factors, initiates adaptive programs to adapt to the new condition. The adaptive reactions are hypertrophy, hyperplasia, metaplasia, dysplasia, atrophy, and others. When adaptive capability of cell is limited, the functional systems are depleted, and this leads to the death of the affected cells. Destruction of the cells and tissues under influence of pathogenic factor is called necrosis. There are several stages of necrosis: paranecrosis, necrobiosis, necrosis, and autolysis. Paranecrosis is the first stage of necrosis and lasts longer than other stages and accompanied by reversible change of cell injury. If favorable conditions occur, the life of cell is restored. Necrobiosis is a short-term process, unlike paranecrosis. Necrobiosis is characterized by irreversible change in the cells, where cell injury is deepened and results in necrosis. Necrosis means complete stop of the cell's life. We distinguish reversible and irreversible change of cell injury, increasing of membrane permeability and formation of blebs on the membrane, swelling of the cells, as well as swelling of mitochondria and endoplasmic reticulum, detachment of ribosomes from endoplasmic reticulum, condensation of chromatin in the nucleus, all are the reversible change. Reversible change don't lead to the cell death if the damaging factor is removed. In this case, disturbance of intracellular homeostasis is temporary. During further aggravation of injury, irreversible change occur: loss of the cell membrane integrity, cessation of ATP synthesis in mitochondria, outflux of the hydrogen ions and cytochrome C from mitochondria, fragmentation of DNA, loss of lysosomal membrane, autolysis of organelles are irreversible change of cell injury. The inevitable consequence of these changes is necrosis. Etiological factors of cell injury may be exogenous and endogenous. Usually the exogenous factors cause acute and endogenous factors cause chronic type of the cell injury. Exogenous factors are mechanical factors, for example, trauma, stroke, wounds, and etc. Physical factors, high and low temperature, ionizing radiation, and others. Chemical factors, acids, alkalines, some medicine, enzymes, their inhibitors, activators. And at last, the biological factors, viruses, bacteria, parasites, and etc. The endogenous factors may be ischemia due to the spasm of vessels, atherosclerotic change, thrombosis, stasis, neurotrophical lesions of tissue, for example, disturbance of innervation in tissue, cessation of innervation, gene mutation and hereditary enzymopathies, for example, sickle cell anemia, thalassemia, and etc., immune, autoimmune, allergic processes, for example, in rhesus conflict, Hashimoto thyroiditis, arthritis phenomenon. Besides of these, hypoxia, energy deficiency, accumulation of the harmful products of intermediate metabolism, acidosis, alkalosis, and other endogenous factors may cause secondary alteration. Aside from necrosis, another form of cell death called apoptosis. Apoptosis is a genetically programmed cell death. Apoptosis can occur both in physiological and pathological states. In physiological states, it serves to clean tissue from unwanted cells that have completed their physiological functions and have lost significance for the body. For example, regression of the breast cells after lactation, apoptosis of alpha-aggressive T lymphocytes during the development of immune tolerance. In pathological conditions, apoptosis ensures the destruction of damaged and harmful cells, for example, killing of viruses infected cells, as well as neoplastic cells by cytotoxic T lymphocytes. These slides show us differences between necrosis and apoptosis. Necrosis occurs in a large group of cells and usually begins from increasing of permeability, membrane permeability, cell swelling, and follows pathological change in the nucleus. The cytoplasmic material, lysosomal enzymes, and all biologically active substances spread into the intercellular spaces. As a result, adjacent intact cells are also damaged in inflammation, and inflammation develops. Thus, necrosis is spread to the tissue area and sometimes to whole organisms. Again, these slides show us the differences between necrosis and apoptosis. Apoptosis occurs in a single cell or in a few cells. Shrinkage of cells is observed. The process begins within the nucleus. DNA breaks down into the fragments, condensation of the chromatin, and karyopyknosis, condensation of nucleus, karyorhexis, splitting of nucleus occur. Then degradation of cytoplasm, loss of intercellular communications, and reducing of cell size are observed. Finally, cells divide into fragments covered by membrane. These fragments are called apoptotic bodies. Each of them includes cytoplasmic material and nucleus fragment. Thus, unlike necrosis, here intracellular material doesn't spread into the extracellular environment and doesn't cause inflammation. During apoptosis, the new markers of the apoptosis are expressed on the membrane. These apoptotic bodies are easily recognized by macrophages and are destroyed by phagocytosis. The molecular mechanism of apoptosis. The activation of enzymes called caspases plays a crucial role here. Caspases are activated by two mechanisms, the mitochondrial or intrinsic pathway and the death receptor or extrinsic mechanism. Both mechanisms require an apoptotic signal to initiate, to induce apoptosis. Deficiency of growth factors, ionizing radiation, action of free radicals, DNA damage, accumulation of misfolded proteins inside cells may be an apoptotic signal in the mitochondrial mechanism of apoptosis. These factors activate the gene p53 that in normal state controls the genome stability. p53 blocks the cell cycle in the phase of G1 and activates the another genes of DNA repair system. If DNA repairing is impossible, the p53 activates the genes of apoptosis, for example, BAC, BAD, BAX, and another. All these reasons lead to the imbalance between proteins and stimulate and inhibits apoptosis. In other words, the proteins that activate apoptosis, BAC, BAD, BEAD, and another increases, whereas anti-apoptotic proteins, for example, BCLX, are inactivated. Apoptotic BAD proteins insert into the mitochondrial membrane and form channels. Their permeability increases and cytochrome C and other mitochondrial proteins escape through these channels into the cytosol. Releasing of cytochrome C activates procaspases into the caspases. The changes occurring after the activation of caspases are similar in both mechanisms of apoptosis. Caspases cleave endonucleases and proteases that degrade DNA, nucleoproteins, and protein of the cytoskeleton. And the end result is the cellular fragmentation and formation of apoptotic bodies. Apoptotic bodies surrounded by membrane are digested by phagocytes. Phagocytes recognize a molecular marker of the surface of the apoptotic bodies. In the extrinsic mechanism, death receptor pathway, external signals are received by membrane death receptors. Many cells express surface molecules called death receptors. For example, tumor necrosis factor receptor, FAS, CD95 receptor. When death receptor binds with their ligands, TNF receptor to TNF, fast to TIV ligand, on the surface of target cell. These in turn activate intracellular death domains, and then procaspases convert into the caspases and trigger the apoptosis. The most important mechanisms of development of cell damage are disturbance of energy production in mitochondria or mitochondrial dysfunction and ATP depletion. The second, formation of the free radicals and disturbance of membrane permeability. Calcium mechanism of cell injury. Change of membrane potential and electrolyte osmotic mechanism of cell injury. Then the protein mechanism, genetic mechanism of cell injury. We will explain some of them in the next slide. Mitochondrial dysfunction and ATP depletion are the main universal mechanism of cell injury. Increasing of cell or tissue or organ activity in the several times higher than its energetic potential causes disparity between energy consumption and energy supply. Diminishing of energy production in the cells and development of energy deficiency leads to the functional impairment in mitochondria. It causes an imbalance in the process of oxidation and phosphorylation. The process of oxidation, anaerobic glycolysis, accelerates, but phosphorylation decelerates due to intensive breakdown of ATP and creatine phosphate. Diminishing of ATP destroys the activity of energy-dependent ion pumps, as well as sodium-potassium ATPase, sodium-calcium ATPase, calcium-magnesium ATPase, and others. All this leads to the accumulation of the sodium and calcium inside the cells and potassium outside. Calcium activates phospholipases, which in turn breaks down membrane phospholipids. The membranes of the cells and the membranes of the mitochondria, also lysosomes and all other organelles, are damaged too. Mitochondrial damage results in the formation of the high permeability channels on its membrane, which lead to the loss of mitochondrial membrane potential and pH change, resulting in the progressive depletion of ATP and necrosis of the cell. On the other hand, the release of cytochrome C and the number of the apoptotic proteins from mitochondria may result in the death by apoptosis. The second mechanism is the formation of the free radicals and disturbance of the membrane permeability. It's another important mechanism of cell injury. The development of this mechanism is associated with formation of a large number of free radicals. This process called oxidative stress. Oxidative stress induces by reactive oxygen species, ROS, which belong to a group of the molecules known as the free radicals. Free radicals are the chemical species with a single unpaired electron in an outer orbit. ROS are normally produced by all cells in a small amount. The action of ultraviolet and ionizing rays, as well as decreasing of activity of antioxidant system, accelerates the formation of the free radicals. Increasing of free radicals leads to the lipid peroxidation of the membranes, to the fragmentation of cellular proteins and DNA. Cell membrane phospholipids mainly consist of unsaturated fatty acids, which are easily oxidized by free radicals. Moreover, as a result of formation of a large number of the free radicals, lipid peroxidation (LPO) reactions occur. LPO products damage the membrane and increase its permeability. The penetration of sodium ions into the cell accelerates the transition of water into the cell too. Thus, the cell swells, its turgor increases, the mechanical damage of the membrane leads to its destruction and loss of integrity. As a result of cell injury, not only cytoplasmic and mitochondrial membranes undergo change, but also the membranes of nucleus, endoplasmic reticulum, and lysosomes change too. Destruction of these membranes leads to the development of irreversible change. Karyolysis of nucleus, stopping the protein synthesis in the ribosomes of the endoplasmic reticulum, exit of the lysosomal enzymes into the cytoplasm lead to the irreversible change. Calcium mechanism of cell injury. An increase in calcium concentration inside the cell plays an important role in the pathogenesis of the cell injury. Calcium may pass into the cell by these three pathways. Calcium passes from outside, from intercellular space into the cell as a result of increasing of permeability of the cell membrane. The second is transition of calcium from mitochondria to the cytoplasm as a result of disturbance of the calcium accumulating function of mitochondria. Then the transition of the calcium in the cytoplasm occurs as a result of endoplasmic reticulum injury, and at last insufficiency of mechanism ensuring the release of calcium from cytoplasm into the extracellular space. Calcium path, sodium-calcium exchange mechanism. All this is connected with the disruption of the relationship between oxidation and phosphorylation in the cell and the development of energy deficiency and the delay of the function of energy-dependent ATPase. The previously mentioned three basic mechanisms of cell injury, energy deficiency, disturbance of membrane permeability, and calcium mechanism, each causing the other one, lead to the development of the vicious circle. And in brief about other mechanisms of cell injury, for example, change in membrane potential and electrolyte osmotic mechanism of cell injury, this associated with an increase in membrane permeability and impaired activity of ion pumps. Accumulation of sodium and calcium inside and potassium ions outside of the cell change the normal electrical potential of the membranes, and differences of potential decreases. This is accompanied by formation of pathological action potential. The protein mechanism of cell injury. Depending on the function of proteins, various disorders occur. Change in the structure of proteins that act as a receptor and provide the cell response to certain signals lead to the impaired cytoreception. And at last, the genetic mechanism of cell injury is associated with the occurrence of the various types of mutations. Mutation of a gene encoding the synthesis of specific enzyme leads to a deficiency of this enzyme and disturbs the metabolism in all organisms. There are a number of protective mechanisms that prevent cells from development of damage. The function of these systems are directed against the main mechanism of cell injury and carried out by the following systems. The first of antioxidant system, you see here. The system neutralizes the free radicals. The antioxidant system enzymes include, for example, SOD, superoxide dismutase, catalase, glutathione system, glutathione peroxidase, glutathione reductase, and etc. Antioxidants of non-enzymatic origin include vitamin E, vitamin C, A, beta-carotene, ferritin, ceruloplasmin, steroid hormones, and others. The next system is anti-mutation system, DNA repair system. This prevents mutations. This system includes the enzymes such as helicase, endonuclease, hexanuclease, DNA polymerase, ligase, and etc. Then detoxification system. This system neutralizes toxins that are cytochrome P450, glutathione S transferase. Then the heat shock the proteins. This system prevents the denaturation of the proteins and their improper package. They take role in the utilization of the misfolded proteins. And buffer system, this system regulates acid-alkaline balance in the body. In the second part of our lesson, as usually, we reproduce the experiment. In this experiment, we will disturb the function of ciliary epithelium on the mucous membrane of oral cavity. For this experiment, we need the frog, the cork planks, scissors, pins, thread, and solution of hydrochloric acid. We take the frog, make motionless by the way of destroying of the spinal cord, and then we fix the frog on the cork plank on its back side. Then we cut off lower jaw, and some small pieces, 1 millimeter of the pieces of thread are put on the mucous membrane of palate. After 3-5 minutes, we observe the movement of these thread pieces to the esophagus due to normal function of the ciliary epithelium. Then the mucous membrane of oral cavity is smeared with hydrochloric acid solution, and again pieces of thread we put on it. As a result of mucous membrane alteration, the motor function of epithelial cells are disturbed, and the pieces of thread don't move. Now, explanation of this experiment, the explanation of the observed change. This experiment demonstrates us the functional change of cell injury. The functional changes are usually earlier than morphological. Besides, we reproduce here the non-specific sign of cell injury. As you know, signs of cell injury we divide in two groups, specific and non-specific signs. Specific that arise, the signs that arise under influence of only one particular factor, are called specific. And non-specific signs are changes that occur under influence of various damaging factors. If we use any other chemical substance here, but not hydrochloric acid, for example, physical factor or the heat, cold, the result will be the same. That's why in this experiment we perform and observe the non-specific sign of cell injury. Now let's do some test questions as we usually do in our lessons. Question number 1. Choose the endogenous causes of cell injury. A. Ultraviolet lights, poisoning, emotional strain. The B. Alcohol, infection, physical strain. All they are false. The right version is C. Denervation, embolia, mutation. D. Ionizing radiation, high temperature, helminths. And E. Decompression, kinetosis, and cavitation. As you see here in the version C, there are only endogenous factors are shown: denervation, embolia, and mutation. Denervation means the lack of the innervation when tissue is lost the nerve supply. Embolia is an occlusion of blood vessels with beads of matter, for example with thrombus. And mutation, you know, the permanent change in genetic apparatus. And all three are these factors are endogenous. All others, ultraviolet light, poisoning, emotional strain, the alcoholism, the infection, physical strain, ionizing radiation, high temperature, helminths, are exogenous factors. And you study about their action in general nosology. And now I would like to remind you the meanings of terms in the version E. Decompression. Decompression occurs in the condition of lowered atmospheric pressure. Kinetosis is the action of accelerated movement on the body, and cavitation is the action of ultrasound into the tissue. All are the exogenous factors. The next question: cell injury leads to A. Hyperkalemia. It's the right version. B. Decreasing of intracellular sodium, decreasing of intracellular calcium, inactivation of the phospholipase, decreasing of free radicals. Now explanation. We say that when cell is damaged, the work of pumps at sodium potassium interface is impaired, and membrane permeability increases. And as a result of the sodium and calcium accumulate inside and kalium outside of the cell. And due to the accumulation of the potassium ions out of the cells, hyperkalemia may occur. That's why the correct version is A, hyperkalemia. And in contrary of the version D and E, during cell injury, phospholipases are activated and free radicals increase. The next question is: increasing of calcium in cytoplasm leads to, first, decreasing of ATP, right, activation of phospholipase, right, activation of proteases, it's right, correct, inhibition of ATPase, it's wrong, and inhibition of endonuclease, it's wrong too. That's why the right version, the correct version is D, 1, 2, 3. And now the explanation. We have just noted that decreasing of ATP, activation of the phospholipase, and activation of the proteases are the chains that are connected with the accumulation of calcium ions inside the cells. And version D, that's why, is right. As shown in 4, inhibition of ATPs and 5, inhibition of endonuclease are false, because cell damage is accompanied by activation of the ATPases and endonucleases, but not by their inhibition. Next question is, mark the non-specific sign of cell injury during cyanide poisoning. A. Increasing of membrane permeability, which is correct. Inactivation of cytochrome oxidase, inactivation of hexokinase, inactivation of cholinesterase, increasing of synthesis of ATP. We say that signs of cell injury can be divided into specific and non-specific. Non-specific signs occur during all cases of the cell injury and don't depend on the nature of damaging factor, but specific signs depend on the type of injury factor and occur under the influence of only these damaging factors. Non-specific signs of cell injury include increasing of membrane permeability, acidosis, edema, protein denaturation, increased activity of number of intracellular plasma enzymes, dystrophic calcification, and so on. These are the general signs of cell injury, and they accompany all the types of cell injury. Please don't confuse here. You know that as a result of the action of the cyanides, the activity of cytochrome oxidase inhibits. This is specific chain for cyanide poisoning. But here you are asked about non-specific sign. That's why the correct is A, increasing of membrane permeability. Inactivation of the hexokinase is a specific sign of the fluorescein poisoning. Inactivation of cholinesterase is a specific sign of the action of the toxic phosphor organic compounds, and increasing of synthesis of the ATP is wrong because during cell damage, depletion of ATP is observed. Then the next question is, mark the specific sign of cell injury during ionizing radiation. A. Swelling of cells. B. Denaturation of proteins. C. Acidosis of cells. D. Activation of proteases. And E. Acceleration of free radical formation. Now here you are asked about specific sign of cell injury, particularly during ionizing radiation. A specific sign of cell injury is characteristic for only one particular damaging factor. For example, a specific sign of burn or exposure from high temperature is protein denaturation. With chemical damage, a chain inactivity of one enzyme is regarded as a specific sign. Specific sign during action of radiation is formation of free radicals in the body. That's why the correct is E, acceleration of free radical formation. Cell swelling, denaturation of proteins, acidosis, and activation of proteases are the general, non-specific signs of cell injury. The next question. Choose a non-enzymatic antioxidants. A. Catalase. B. Vitamin E, vitamin C. C. Glutathione system. D. Salt, superoxide dismutase. And E. Cytochrome P450. As we note above, antioxidant system includes the substances of enzymatic and non-enzymatic origin. Enzymes of antioxidant system are salt, superoxide dismutase, the catalase, and glutathione system. Antioxidants of the non-enzymatic origin include vitamins E, vitamin C, vitamin A, beta-carotene, ferritin, ceruloplasmin, ubiquinone, steroid hormones, and others. That's why the correct is B, vitamin E and vitamin C. And explanation for the last question. The question is, select the main principle of the intracellular detoxification, the principle of this system. A. Transformation of the hydrophobic substances to the hydrophilic form. Then B, prevention of peroxide oxidation of the lipids. C, elimination of the injured DNA. D, protection of cells from the action of acids. And E, protection of cells from the action of free radicals. We know that the detoxification system ensures the neutralization and the removal of the toxic substance from the cells. This function is carried out with the participation of the enzymes. These enzymes, cytochrome P450, the glutathione S transferase. The role of detoxification system to convert hydrophobic toxins into the polar compounds. By this way, they neutralize and remove them from the cells. When substances pass from the hydrophobic form to polar, they have a less toxic effect on biological membranes. In the polar state, these substances easily dissolved and removed from the cells. For example, cytochrome P450 accumulates in the endoplasmic reticulum of liver cells, lungs, skin, and they oxidize non-polar compounds and remove them through the hydrophobic zones or biological membranes. That's why the correct version here is A, transformation of the hydrophobic substances to the hydrophilic forms. Version B, prevention of the peroxide oxidation of lipids. It is the action of antioxidant system. Version C, elimination of the injured DNA. The action of is the main function of anti-mutation system. In version D, you see protection of cells from the action of acid is connected with the action of buffer system. And in E, protection of cells from the action of the free radicals is again the function of antioxidant system.
There are several types of microcirculatory disorders: intravascular disorders, acute during disorders of laminar flow and rheological properties of the blood, disturbances in the blood coagulability, aggregation and adhesion of blood cell, sludge, stasis. What means of sludge? Sludge is blood cells are stuck together and plasma is separate. Stasis is blood flow is stopped. Vascular disturbance. In this case, the vessel wall is damaged because this process and occur during increasing of permeability of the vessel walls. Extravascular disturbances occur during edema, scar, tumor, growing outside of vessel and press them and cause disorders in the microcirculation. Next, disturbance of mixed character. You know, a capillary consists two parts, an arterial end and a venous end. Very interesting slide. A capillary consists two end. You say about it, arterial end and venous end. Fluid from the arterial side of the capillary bed move into the interstitial space and is reabsorbed in the venous side of the capillary bed. That's why fluid is removed during filtration, fluid is absorbed during reabsorption. When this process is disturbed, the filtration and reabsorption process is disturbed. You see arteriole and venule between the lymphatic system. Lymph circulation insufficiency are distinguished: mechanical insufficiency, dynamic insufficiency, resorption insufficiency, and a mixed type. Mechanical insufficiency have two types: organic disorders and functional disorders. Organic disorders, compression of the lymph vessel by tumor scar. Functional disorders develop as a result of spasm of lymph vessel and increase of venous pressure. Dynamic insufficiency, when a large amount of fluid passes into interstitial space and the lymph vessels are unable to transport them. Resorption insufficiency develops as a result of change in biochemical property of tissue proteins. A large amount of proteins accumulated into interstitial space and the lymph vessels are unable to transport them. Next, mixed type. When the transport of fluid through the lymphatic system is disturbed, occur edema. There are chronic, local, general, and acute edema. Tissue volume is increasing and structure of skin is changed in edema. Elephantiasis develop following by this mechanism. Skin of extremity became look on elephant skin. The type of microcirculatory disorders: arterial hyperemia, venous hyperemia, ischemia, stasis, thrombosis, embolism. Arterial hyperemia is an increased content of blood supply to an organ or tissue due to increasing blood flow through the arterioles. Normal capillary bed is the arterial hyperemia, dilated of vessel, increasing in flow. This process develops in the inflammation. According to the pathogenesis, neurogenic and myogenic arterial hyperemia are distinguished. Neurogenic mechanism consists of mechanism, neurotonic mechanism, and neuroparalytic mechanism. Neurotonic mechanism, activity of the parasympathetic nervous system increases, and myotonic mechanism, neuroparalytic mechanism, and activity of the sympathetic nervous system decreases. Myogenic mechanism, the cell is damages. The damaged cell secretion biological activity substance. They dilate the blood vessel. Types: arterial hyperemia, physiological arterial hyperemia, pathological arterial hyperemia. Physiological arterial hyperemia occurs during functional hyperactivity of organ and tissue. Pathological arterial hyperemia occurs during bones, inflammation, nerves, and endocrine disorders. Physiological arterial hyperemia distinguish working hyperemia, reflex hyperemia, and conditional reflex hyperemia. Working hyperemia is a result of increased activity of organ and tissue. Reflex hyperemia, increase of blood supply of skin under the influence of physical and chemical stimuli. Conditional reflex hyperemia, reddening of the face in shame. Pathological arterial hyperemia distinguished angioneurotic hyperemia, occurs as a result of increased tonicity of vasodilated nerves or decreasing tonicity of vasoconstrictive nerves. Postanemic arterial hyperemia, fluid accumulated in the cavities of the body. If fluid accumulated in the abdominal cavity and form ascites. Next, if fluid accumulated in the pleural cavity, maybe form a hydrothorax. After is rapidly pumped out. Collateral arterial hyperemia is a result of occlusion of artery. Its proximal branches are dilate. Acute arterial hyperemia is resulted from decreased pressure on a body surface when Caisson worker or divers are rapidly lifted. Inflammatory hyperemia. Next, hyperemia connected with artery venous anastomosis. Arterial blood passes into veins through anastomosis. Signs of arterial hyperemia: redness of skin and mucous membranes, increase of tissue tension, accelerate of the blood flow, temperature elevated, increasing in the number of functioning capillaries. Outcomes: leads to improved metabolism, increase of the blood supply to brain, rise of the local arterial pressure. Venous hyperemia is blood supply increases in organ and tissue, and blood circulation diminished in organ as a result of impeded outflow of the blood. Normal capillary bed. In time, dilation vessel. This process develops in the congestion and maybe venous hyperemia or congestion hyperemia. Develop cyanosis, hypoxia, decreased outflow, and there are local obstruction, congestive heart failure. Causes venous hyperemia: cardiac insufficiency, If right ventricle failure causes venous hyperemia in the greater circulation, if left ventricle failure causes venous hyperemia in the lesser circulation. Veins are occluded by thrombus, veins are compressed by thrombus and scar, capillary permeability increases and filtration increases. The wall of the veins genetic weakening. Signs of arterial venous hyperemia, cyanosis of skin and mucous membranes, expansion of vascular lumen, stasis, temperature decreases, blood flow decreases, and from hypoxia, circulatory and histotoxic, formed hypercapnia. Outcomes: venous hyperemia, hypoxia, acidosis, edema, dystrophy, fibrosis, and etc. Development of ascites. Next, formation of thrombus. Ischemia, blood supply decreases of blood and tissue. As a result of arterial blood inflow reduces. Type of ischemia: spastic ischemia, compression ischemia, obstructive ischemia, and redistributes ischemia. Compression ischemia, compression of the vessel by tumor, and obstructive ischemia and clot, emboli obstruct the vessel. Next, signs of ischemia: colorless of tissue and organ, blood flow decreases, arterial pulsation decreases. Outcomes of ischemia: hypoxia, acidosis, necrosis of tissue, and infarctus. Stasis is complex causation of the blood flow in the microcirculatory system's valves. There are three types of the stasis: ischemic stasis, venous stasis, and capillary stasis. Ischemic stasis is resulted from causation of blood inflow from arteries into the capillary. Venous stasis or congestive stasis is resulted from causation of blood outflow from capillary to the venous system. Capillary stasis or true stasis is independent circulatory disorders in the microcirculatory system. Stasis is a reversible process. Blood flow may be restored after elimination of stasis cause, but it causes vascular walls damage and erythrocytes adhesion in the vessel, as develop irreversible stasis process. Infarct is necrosis of tissue as a result of sharp disturbance of the blood circulation. There are two stages: ischemic stage and necrotic stage. Have type. In fact, have several types: white infarct, red infarct, and white infarct surrounding by the red border, and white infarct absorbed in the kidney, in the spleen, and red infarct absorbed in the lung system, in the intestine. White infarct surrounding by the red border absorbed in the heart and kidney. Outcome of infarct may be from coagulative necrosis and colliquative necrosis. Coagulative necrosis develop in organ which has more protein and calcium, and may be developed in the cardiac muscle and kidney. Colliquative infarct develop in organ which has more lipids and water, and develop in the brain. Thrombosis is formation of blood clot in the blood vessel or cavities of the heart, with the result that they impede the circulation. The main cause of thrombus formation: three factors are required for thrombus formation, and endothelial injury after abnormal blood flow. Next, hypercoagulability. Stage of thromboformation have three stages: vascular stage, thrombotic stage, and plasmatic stage. Vascular stage, vascular wall damage lead to the vasospasm. From damaged cell leak out biological activity substance that change of the platelet properties. Next, thrombocyte stage is the damage area of the vessel wall, platelet accumulated, and platelet come to the damaged area. There is a Willebrand factor in the damaged area. Willebrand factor and hair bind to the receptor on the platelet. If Willebrand diseases occur in the deficiency of Willebrand factor. There are other syndrome, Bernard-Suller syndrome. In the Bernard-Suller syndrome occurs in the deficiency of platelet receptor. In this case, platelet adhere to the damaged area. This is called platelet adhesion. Platelet adhesion. When the platelet approach each other and aggregation in the damaged area. If the platelet aggregate is impaired, may form Glassman thrombocytopenia, Glassman diseases observed. Willebrand diseases is transmitted to the generation by autosomal dominant way. Bernard-Suller and Glassman diseases is transmitted to the generation by autosomal recessive way. Next, plasmatic stage. Plasma clotting factor are activate, and clotting system consists of internal and external pathways. External and internal pathway together activate the tense coagulation factor. After activate prothrombin. Prothrombin is converted or turned to thrombin, into thrombin. After fibrinogen is converted to fibrin. Eventually a clot forms. The types of thrombi there are under white thromb, red thromb, mixed thromb, hyaline thromb, small thromb, and obstructive thromb. White thromb consists of leukocyte, platelet, and fibrin. Red thromb consists of leukocyte, platelet, fibrin, and erythrocyte. And hyaline thromb consists of decomposite blood cells and precipitated plasma protein. Outcomes of thrombosis, aseptic autolysis, occurs in the small thrombi. Septic autolysis occur under influence of pathogenic bacteria and thrombo breakdown with spreading of infection in all organisms. Organization, canalization, and vascularization of thrombi replaced by the connective tissue organization. When the canal form canalization, blood flows through the canals, vascularization. Detachment of thromboembolism develop of infarctus. If accumulation, calcium accumulation in the thrombus causes petrification, and stone accumulation in thrombi causes phlebitis. Outcome of thrombosis, and a very interesting slide, resolution thromb, embolization of to lung system, organizate and incorporate into wall, organizate and recanalizate this. Embolism is occlusion of blood vessel with beads of matter carried by the blood or lymph and usually falling to the bloodstream. Emboli are divided into two groups, endogenous embol and exogenous embol. Endogenous embol, thrombi, fat, tissue, cell, amniotic water, and the exogenous embol, air, gas, solid foreign bodies, bacteria, parasites. The types of emboli air emboli, formed during damage of large vessel, gases emboli, and formed during in the Caisson disease in divers, thromboemboli acute during varicosis of veins, atherosclerosis, fat emboli acute during fracture of bones, and etc. cellular or tissue emboli, bacteria emboli, and foreign body emboli, and etc. Emboli move in three main directives: emboli circulatory system, emboli lymphatic system, emboli of the great circulation, emboli of lesser circulation, of the portal emboli. Let's look at the test to better understand the topic. Test, and the correct answer are variant A. Choose the intravascular type of microcirculatory disorders. And right and 5 answer. Disturbances of rheological property of blood, damage of vascular endothelia, increasing of prostaglandin synthesis, degranulation of mast cell, increasing of vascular wall permeability. And true answer is A, disturbance of rheological property of blood, because asked intravascular disorders and rheological property. Next slide. Choose a version with correct type of lymph circulation pathology. Type, and there are written on our slide. Written in our slide, and type of lymph circulation: mechanical and dynamic resorption. Next slide. Choose the reason of venous hyperemia and compression of the venous with connected tissue from the outside. Venous hyperemia or congestion hyperemia, formation stasis, and through answer A. And other variant, write about arteria, but A variant, write about venous vessel. Next slide. 4. Which type of microcirculatory disorders correspond with the following: accelerate of blood and lymph flow in microcirculatory system, increasing of volume of tissue and organs, elevation of the temperature, and redness. So answer and A. Why? Because in the arterial hyperemia, increasing blood inflow, increasing temperature, increasing and volume of tissue and organs. Because, and choose through variant A. Next test, and choose a version with all types of stasis. Type of stasis are written on our slide. Ischemic type, venous type, and through capillary stasis. Next slide. Choose a version with all type of infarct. White infarct, there are white infarct, red infarct, white surrounded by red border infarct. Next slide, and very test, and very interesting test. Which of them take part on the pathogenesis of thrombosis? And is the thrombosis injuring of vascular wall? Next, decreases of velocity of blood flow, decreases blood flow. Next, activate of coagulation system, because coagulation system activate and inactivate anticoagulation system. We choose and through answer A. Laboratory study. Let's read the experiment on this topic. During the experiment, microcirculatory disorders were observed in animals such as rabbits and frog. Microcirculatory disorders were observed in rabbits' ear and frog tongue and frog's mesentery. When we look on the microscope, arterial hyperemia in the tongue is observed. Blood circulation is observed under the microscope in the web, tongue and mesentery. If 1 and 2 drops of several medical solution is right laboratory is poured on the web, microcirculatory disorders is observed. I put it the video, and I won't read the laboratory, and reproduction of the arterial hyperemia is in Frax Tonger, and venous hyperemia in rabbit's ear, and ischemia, compression ischemia in the rabbit's ear, and reproduction of ischemia in the Frax mesentery and verb. Next, and reproduction of white thrombus in Frax mesentery vascular wall and white thromb, and in time and frog, the Frax central nervous system is destroyed after fixed on the plaque, and abdominal skin, muscles, and peritoneum are cut. Next, small intestine is carefully put out and fixated over the hole, and several over the hole, and under the microscope, the normal blood circulation in the mesenterial vessel is observed. Several minutes later, when mesentery is dried off a little and blood flow is slowed down. A small crystal of sodium chloride on the end of needle is put beside that place, and few minutes after formation of white thrombus. I put it the video after reproduction of fat embolism in Frag blood vessel, and primary Frag's central nervous system is destroyed after thorax cavity is opened, and 1-2 milliliter of Vaseline oil is injected to the ventricle of heart system. Later, and the frog's tongue fixation over the hole, and under the microscope, movement of the drops of oil in the vessel of tongue or mesentery, and abstraction of the vascular lumen by the fat embolism are observed. I won't show the video. Blood vessels are damaged, blood pours out. This process is called bleeding. Next, oil embol moving in the vessel. And with venous hyperemia. Next, formation of white thrombosis in the Frax mesentery. Thanks for your attention. Take care.